Okay, welcome to Lipids Day 2. These are all of the learning objectives for the entire video. So by the end of this, you should be able to define key vocab terms, draw a tag or triacylglycerol from symbolic notation, draw the mechanism of tag esterification. This is also known as tag formation, which is a type of reaction that's called a condensation reaction. Compare the functions of energy storage, membrane lipids, egg signaling lipids based on their structure. You'll need to refer back to the other lipids video to help you with this. And then describe the functions of steroids and icosanoids and compare the functions of steroids and icosanoids based on their structures. So just as like a kind of a recap and also a where are we going with this, I made this little diagram to sort of help you, organize your lipids structure function information. So what's at the top is, is the fatty acid. So if you'll remember that all lipid structures, all types of lipids start with a fatty acid structure. And the ones that we've seen so far, I'll switch to different color. The ones that we've seen so far are sterols and the phospholipids. And again, these are generated from fatty acids. So the sterol has that steroid nucleus with the A, B, C, D, and then it has the alcohol, and then the rest of it is just carbon-hydrogen, carbon-carbon bond. And the phospholipid has the glycerol backbone, which is this piece right here. The two fatty acid tails, that's this piece here. It has one phosphate, that's this piece here, and then one functional group here. And there are three other kinds of lipids that we're going to be talking about today. So we're going to be talking about triacylglycerols, and their primary function is energy storage. Then we're going to talk about icosanoids and steroids, and both of these are signaling types of lipids. And the way that I've organized this is that if you're looking at this going from left to right, the triacylglycerols, or what's known as the tags, are the most hydrophobic of the lipids. And then if you go all the way to these are the least hydrophobic. I'll just do that one. The other way that we could write this is we could call these are the least polar. And over here are the most polar. Or the least hydrophilic. And these are the most hydrophilic. And that's because of their structures. And then that also plays into their functions. So let's go ahead and get started with the tags. Okay, so triacylglycerols, TAG, the abbreviation are tags, are your primary fuel or energy storage. So when you need to store any like dietary fat, these will be stored as tags. So let's say you eat, let's say you eat a stick of butter. I don't know why, but let's say you do it. You eat a stick of butter and you don't need all the energy from the butter, all the calories essentially from the butter, then it will take all the fatty acids from that butter, which are probably saturated fats because it's a solid, and it will incorporate them into a tag instead of letting them be free-floating fatty acids in your body. Your body can also make fatty acids for storage, and then those will go also to a tag. So let's talk about the structure first, and then we'll kind of like bounce back and forth between structure and function. Okay, so two versions of essentially the same thing. So tags have two parts. This part that's up here is the glycerol backbone. And then these down here are the acyl tails. So hopefully this looks really similar to the phospholipids, except for instead of on the third carbon there being a phosphate, there's just another tail. So we've got 3 tails that were each once a fatty acid, and those are all attached to the glycerol backbone. So there's 1, 2, 3 carbons of the glycerol backbone, there's their oxygens, and then the rest of it was the fatty acid. And then this view is the spheres view, just showing like the relative size of each of the atoms colored in CPK, so you can see sort of this like polar-ish head group. It's the least polar of all the head groups because there's no charge. There's no negative charge, there's no positive charge, there's nothing extra sticking out of it. In fact, a lot of times, unfortunately, I don't really like this, but a lot of times tags are considered neutral, and that's just in terms of charge, meaning they don't have a charge. Overall charge here is 0. So they don't go into membranes because they don't make all those favorable interactions with water. But because of their structure, they do have a very important function. They sequester all the negative charges of the fatty acids so that you can get a lot of fatty acids in an itty-bitty living space. Okay, what am I talking about? So if you were to, think about that stick of butter, it's saturated fatty acids. At pH 7, they're going to be negatively charged. I'll do this one this way to just try and make a point. Whoa, yikes. If you were to try to store all these fatty acids, you'd have a lot of charge repulsion because you'd have a lot of negative charges that you're trying to fit in a small space. And that means that these would push on each other, and you would need to get a much bigger space to fit all the fatty acids. So, trees are so big because they store carbohydrates, and carbohydrates are huge. And so trees just get bigger and bigger and bigger and bigger because they have more and more and more carbohydrates to store. Well, if you were like a tree, we'd all have to get bigger and bigger and bigger and bigger and bigger and bigger and basically be immobile if we've stored all of our fatty acids as fatty acids. So instead, the body moves them to make tags so that the tag, is that not what I thought it was? Is that an eraser? How about you? Are you an eraser? Yeah, there we go. So that the tags up here in this glycerol backbone don't have any negative charge, and then you can put a whole bunch of tags together. Okay, so let's draw the mechanism. I'm just going to pause for a second. Okay, I didn't leave myself a blank page, so I'm just going to I'm going to erase some things. And if you don't yet have a decent sized piece of paper to draw on, get some paper and some stuff to draw with, because I'm going to scooch all of this out of the way. Scooch it up and away. Goodbye. Okay. All right, I'm going to start with, so remember the pieces to make a tag are you need glycerol, and glycerol is 3 carbons, and I always draw mine like this. So here's 1 carbon, 2 carbon, 3 carbon. O, I'll draw this H kind of kinked out to show the lone pairs there, and then I'll just draw these other 2 OHs. So you need glycerol. And then you also need fatty acid. So I'll draw that in a different color. I'll do pink. Maybe I'll do green. All right, so I've got my glycerol, I've got my fatty acid. So we're gonna draw the condensation reaction, which is also called, in this case, esterification. Whoa. Which literally means to make an ester. bond, which if you remember should look something like this. And in the process, because it's a condensation, we need to make or eliminate water. Okay, the fun thing about biochem is that everything is in water. So I'm just going to add a water over here to start to make a good leaving group. So when you look between these two molecules, what you'll see is that I like to draw some of those partial charges. So here I've got, arrow points the most electronegative, points here. So I've got a fairly strong partial positive on this carbon here. And then when I look at glycerol, if I'm just trying to figure out like who the nucleophile is and who the electrophile is, arrow points here, arrow points here. So I've got a pretty strong partial negative here and not as strong of partial positives. So to me, what that means is that this oxygen here is going to be my nucleophile, and this carbon here is going to be my electrophile. But what's attached to this electrophile, neither of these things are good leaving groups. So let's make a good leaving group. And because everything's in water in biochem, we just get to use water sort of egregiously. And one of the best leaving groups is also water. So let's start to make a good leaving group. So I'm just going to take this hydrogen off of a nearby water. I'll put this here. There's a lot of waters around and H3O pluses. So I don't worry too much about where that first hydrogen comes from because there's water around. Okay, so I put one hydrogen on. Then I'm going to get another hydrogen, and I'm going to take it from here, and I'm going to put electrons there to make that oxygen even more a better of a nucleophile. So what do I have so far? So what I've got so far now is I've got an O minus, really good nucleophile. And I also have I've got a good leaving group, and I have a good electrophile with this partial positive of this carbonyl carbon. And now I can do the attacking part where the negative electrons move towards the positive atom. Electrons go up, electrons come down, and we're going to take the electrons from this bond and drop them onto that water oxygen, and that kicks out the leaving group. So then what I have from here is the O, the glycerol backbone, still intact. I made a bond to the carbonyl carbon, so I'll just do that carbon in green because it's this carbon over here. This oxygen, this carbonyl, is still from the fatty acid, and then the tail is still there, and what came off is water. So we did one condensation reaction, which in this case did an esterification to make an ester bond. That's this bond right here, made this ester bond. And now we've attached one of our fatty acids that had a negative charge onto the glycerol backbone. So it takes out the negative charge, but you don't lose the fatty acid tail, which is where all the energy is stored. And essentially, this just goes on 2 more times to attach tails here and here. So you'll release 2 more waters and need 2 more fatty acids to get all the way to a tag. And if I were to give you something like, I don't know, ask you to draw, I don't want that. Go back, go here, let's do this one. To draw fatty acid, I'll just say condensation, to a tag with, I don't know, let's say that they're all the same. 3 fatty acids that have this structure. 18:1 trans delta 8. Then each of the tails will have this structure. So each of the tails, you would count, this would be carbon 1, 2, 3, 4, 5, 6, 7, 8. Throw in the double bond. 9, 10. This is 9, 10, 11, 12. And then you'd have to extend it. 13, 14, 15, 16, 17, 18. So that would be how you would draw a tag given the symbolic notation of the fatty acid. Cool. All right. So let's go back to, again, just as a reminder of structure, function, structure, function. So remember the tags, their function is to hold energy. They are the fuel storage. And in order to be able to do that, you need to get a lot of storage in a small space. So in their structure, they remove the negative charge so that they fit inside of what's called a lipid droplet. And a lipid droplet is just a really fancy way to say a vesicle that holds tags. So if you'll see, it's a vesicle because it's a monolayer. A monolayer, so that type of tennis ball. And then what's on this inside are all the tags. And then the reason they're on the inside is because of the hydrophobic effect. So the tags are the most hydrophobic or the most nonpolar or the least polar, however you want to say it. These are hydrophobic molecules, relatively hydrophobic molecules, so they'll go on the inside of the vesicle to be stored. And when tags go on the inside of a vesicle, it's just called a lipid droplet. And so I just wanted to show you just kind of like some different renderings of lipid droplets based on adipose tissue or fat cells. So there's really like 3 main kinds of fat cells. There's the white, which is what you have this is the primary amount or primary type of fat cell that you have, especially as an adult. Beige, which is more uncommon, and then brown adipose tissue, which you have as an infant, and then you don't express any more of it when you get older. And so what's on the inside are these right here are the lipid droplets. This is just showing where all the tags are stored, and they're stored in one big giant vesicle in the center, and it sort of pushes, for the white adipose tissue, it pushes things like the mitochondria and the nucleus to the outside. And the beige and the brown adipose tissue just have more mitochondria for thermogenesis, and their lipid droplets are tinier. So this is just a more interesting image of a brown fat cell. And so you can tell that it's a brown fat cell because there's a whole bunch of lipid droplets. And then there's also a ton of purple mitochondria. And of course, there should always and only be one nucleus. So again, structure, function. Phospholipids and sterols would make very poor fuel storage. And you'll see that the next ones are also poor fuel storage, but are great for other things. And we're going to talk about signaling next. Okay, so this is just a really I don't know, I just like this figure. Brief recap of your storage lipids. See, this one even says that they're neutral, and that just means in terms of charge. Triacylglycerols have the glycerol backbone and 3 fatty acid tails, whereas the membrane lipids, which are polar, meaning in this case they have some sort of charge on them, have also the glycerol backbone or the sphingosine backbone, and then 2 tails. 1, 2, 1, 2, 1, 2, 1, 2, and then what's in blue is the piece that makes them really amphipathic or amphiphilic and able to form the vesicles and the lipid bilayers, whereas a fatty acid does not have this really polar phosphate functional group or saccharide functional group. Let's move on. So I'm going to do icosanoids first. Icosanoids are local signaling molecules. Most people kind of panic a little bit when they see the word icosanoid. And the other word that I think that you may have seen that starts with an EI is Einstein. And so they both start with the same I sort of sound to them. So Einstein, icosanoids. Icosanoids are local signaling molecules. So I'm starting with a function on these ones. And I picked a couple different, situations that are sort of local. So whenever you get stung by a bee, you get local redness and swelling. You don't get swelling unless you have anaphylactic or a global allergy to a bee sting or a mosquito bite, or think of any kind of insect bite. Most people just get some localized redness. So it's just right around where the bee sting or the mosquito bite is. If you're having some kind of pain in your body, it's usually in one part of your body, one spot. And this person just has some kind of back pain. Maybe it's your elbow, maybe it's your knees, but it's local, right? Same thing. The key word here is local. Swelling is another one. So swelling is also tends to be local, like maybe your feet swell or your hands swell. I don't know, this one's like got a whole leg and a whole arm. This is the best picture I could find. But swelling also tends to be local or can be local. And then another thing that happens in your body is fever. And fever is also tends to be local or just at the site. And these kinds of bodily reactions to something that is happening to you are caused by icosanoids, because icosanoids are your local signaling molecules. And there are 3 kinds of icosanoids. One type of icosanoid is called a prostaglandin, and the prostaglandins always have the abbreviation PG and then something that follows it. A second type of icosanoid is called a thromboxane, and these always have TX as an abbreviation and then something that comes after it. And then the third type are the leukotrienes, which I forgot to put the abbreviation. The leukotrienes always start with LT and then have something that comes after it. I believe this one is LTA4. Okay, so again, when something local is happening in your body, like you were chopping vegetables and you cut your finger, you're going to get swelling and redness right there. You run into a doorknob or something, you're going to get some kind of swelling right there at the site of where this is happening. And that's all caused by these icosanoids. And the reason that they're local signaling molecules is because of their structures. And so I'm going to move to the next slide so we can talk about their structures and how you recognize them, but also where they come from and how that influences their function. Okay, so icosanoids all come from this molecule called arachidonic acid. And arachidonic acid, I'm just going to zoom in here. arachidonic acid is a membrane lipid, and it comes from one of the tails of a phospholipid. So if the phospholipid tail is cut off and it's arachidonic acid, which is this structure over here, then that can be turned into any of the icosanoids. And the reason that this is important is because all cells have arachidonic acid in their membrane, which means that any cell can make this like local signaling event happen, right? So it doesn't matter if you bump your head or your knee or your back or your toe, if there will be arachidonic acid in the membranes of those cells, and so you will get that local pain and inflammation at that site because arachidonic acid is there. And arachidonic acid is a fatty acid. It's a particularly special fatty acid. It's 20 carbons long. So if you were to count this, it's 20 carbons long. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 carbons long. And that's important because Icosa equals 20, or is the word for 20 in Greek. So the icosanoids all come from a 20 carbon fatty acid, and Icosa is 20 for Greek. And it has the cis double bonds, and you could probably do the symbolic notation for it, but I'll let you do that at another point. Okay, so from arachidonic acid, you can get to all these ones that start with LT, 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 that one's an LX. So these are the leukotrienes. And what you'll notice about the leukotrienes in comparison to arachidonic acid is that they all just have more OHs or O's on them. They still have a tail or some kind of hydrophobic-y region, but they have more oxygens on them than arachidonic acid. So that makes them more polar than arachidonic acid, and therefore they can float around locally in the watery areas to cause a signaling event with nearby cells. They can't travel too far because they're not polar enough. They're not hydrophilic enough to move through the bloodstream, but they're polar and hydrophilic enough to travel locally because they just have a few more oxygens on them than arachidonic acid. And the same is true for the prostaglandins and for thromboxane. They all also have more oxygens on them than arachidonic acid. All of these are oxygens, 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 oxygen, 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 oxygens. So all of the icosanoids have more oxygens than the fatty acid, and that makes them more polar, more able to move around in the watery areas just for local signaling, but they're just not quite polar enough because they still have all of these hydrophobic regions, so they can't travel too far away. Okay, so how do you tell the difference between a prostaglandin, a thromboxane, and a leukotriene? There is a structural pattern. Maybe you've already seen it, but we'll start with the prostaglandins. So the prostaglandins, if you remember, are all the ones that start with the PG in terms of their abbreviation. All the prostaglandins have a 5-membered ring. All of them have a 5-membered ring. That is something that is unique. This one has 2 of them. And then coming off of the 5-membered ring are these, what to me looks like goofy frog legs. 2 frog legs, 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 2 frog legs. These frog legs are really weird, but they all have this 5-membered ring that has at least one oxygen attached to it, and they have 2 frog legs. So that's how you recognize a prostaglandin. I'm not going to ask you to draw these from memory, but I do want you to be able to recognize them. The thromboxane is just slightly different in that it has a 6-membered ring instead of a 5-membered ring, but it also has 2 frog legs. And then the leukotrienes have no rings whatsoever. They have more oxygens on them than arachidonic acid. They have double bonds, but they all have what I sort of have highlighted in blue. They all have sort of a tail to them, or they like retain that tail. So the leukotrienes, no rings. The prostaglandins have 5-membered ring, and thromboxanes have a 6-membered ring. And just to fully reiterate, remember that their function is to send a local signal. This could be something that's related to pain, inflammation, which would be swelling, redness, or heat. And the reason that they're local is because they're only slightly, missing a T in there, slightly more polar than the fatty acid, but not polar enough to move through the bloodstream. So they just stick out, they just hang out locally. It's like a teenager with no car. You can only get so far on a bike. You got to stay sort of local. And then, send the signals for what you're sending signals for. Okay, those are the icosanoids. So now we're going to do the last signaling lipid molecule, and actually the last type of lipid molecule, and those are our steroids. So in comparison to the icosanoids, steroids are global signaling lipids. And what I mean by that is that they're made in one location in the body, and then they travel. Like this one, they travel to another location or other locations in the body. And the one that I picked just as an example is cortisol. So cortisol made in the adrenal glands and then can travel to really anywhere as a signaler in the body. And there's a lot of steroids, and they have a lot of functions. They're not all made in the adrenal glands, but they do all move through the body. And the reason that they move through the body is because of their structure. And they all have some things that are in common. So all of the steroids have the steroid nucleus, that A, B, C, D nucleus, if you remember from sterols. So they all have a steroid nucleus, but they just have more oxygens on them. Than cholesterol or the other sterols. So the sterols only have one OH, and it's usually down on this ring. But if you'll notice it, cortisol has another O up here, another O here, another O here, another O here, which makes them, again, more polar or more hydrophilic. And because of their smaller compact size, so they have a smaller compact size, they can travel through the bloodstream. Basically, these are the teenagers that have cars, or another creature that gets a car that can move far away places because of its structure, its smaller size, and all of its OHs. make it more easily able to travel through the bloodstream. What the heck is it about the OHs and the oxygens in general that make this able to travel through the bloodstream? Wonder, while I erase, if you can think about what is it about having more oxygens means that it's more able to travel through the bloodstream. What color do I want? Let's do pink. It's because the more non-covalent interactions you have with water, the more soluble in water the molecule is. So the more oxygens, the more favorable non-covalent interactions with water. I'll just start drawing dot 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 dot. And the more soluble in water, the further you can travel through the bloodstream because the bloodstream is mostly water. Cool. So just as, I don't know, again, I don't need you to memorize any of the structures of the steroids, but I do want you to be able to recognize key features of the steroids. So the steroids actually start, well, we start with a fatty acid. And a fatty acid can be converted into cholesterol through metabolism in the body. And cholesterol, this type of sterol, because there's only one OH down here, is the starting point for all of steroidogenesis, or all of the steroids. And the only thing I want to point out about this particular figure is that everywhere along the way, wherever there's like a white bubble is where the oxygen has been added. So it just continues to add on more and more and more and more and more oxygens, and almost every time you do, you get a brand new steroid. That's all I wanted to point out, is that the steroids, they start with cholesterol and just add sequentially more and more and more and more oxygens. And that helps them become more hydrophilic so that they are more soluble in the bloodstream and they can cruise through the body. Okay, that's the end of the second video. And again, this is just a recap of the 5 different kinds of lipids. And those are triacylglycerols, sterols, phospholipids, icosanoids, and steroids. And they all have different functions, and that's because they all have different structures. And remember, these are in order from most hydrophobic to least. And that also plays into their functions. Okay, I'll see you in class. Bye.